Can everyone see week 7 evolution? Thumbs up. Cool, perfect. All right, so let's define evolution. The simple definition is that there's a change over time, right? It's a gradual development of something, of a trait. And then it's oftentimes a different or more complex or better form of that trait when we're talking about species. So again, simple definition is change over time. The biological definition is a change in the genetic composition of a population or species as a result of selective and stochastic pressures over time. So we learned last week that the environment is full of different parameters, abiotic and biotic interactions. How species respond to those parameters and those interactions determines what kind of pressures they have on them. And so thus, as species evolve, we know that all species has derived from pre-existing life. So species aren't the only ones that evolve, right? We see music evolves. Music today is very different from music 10, 20, 100 years ago. We see fashion evolves, right? We see that populations change. Phones, they change over time as well. Here's a graphic of phones from the late 80s, 90s, all the way up into today. We can see it's very, very different. Species and populations are no different. They evolve over time as well. It just happens to be that the time scale which they evolve is hundreds of thousands and millions of years. So it's not as a very short time scale as some of these other things like music and phones and technology and fashion. You can kind of think of evolution as a continuum. So here's an example. We know that the color changes from blue to red, but we can never really pinpoint exactly where that occurred. We see that there's kind of this area of purple right in the middle, but there's not a clear and distinct cut from blue to red. And so we know that from the fossil record that animals have evolved and their traits have changed over time, but we can never pinpoint exactly when that trait and when that change occurred. It's definitely a slow and gradual process, which is why you kind of have this purpley or gray area in the middle. The guy who came up with the idea of evolution is this guy, Charles Darwin. I'm sure you guys have all heard of him before. He did his main conclusions from evolution through working in the Galapagos Islands. Pause my phone here. And working with finches. He sailed on the boat called the HMS Beagle. And there's 3 main ideas through his theory of natural selection and evolution, is that natural variation exists, traits are passed from parents to offspring, and this idea of survival of the fittest. And so these 3 main ideas can be boiled down to descent with modification. Descent with modification includes these 3 main ideas that Charles Darwin came up with. So let's take a look at them. So 1, the idea of natural variation exists. Right, we have a mother dog and her 5 pups, right? 4 of them are going to be white, 1 of them is going to be black. That just occurs naturally. They're all the same species, but there is natural variation that occurs, right? We are all human species, but we do look very, very different. So there's just natural variation that occurs in different types of species, right? Traits are passed from parent to offspring. If you've got blonde hair, most likely your parents have blonde hair. If you have blue eyes, most likely your parents have blue eyes, right? There's just inheritance is a thing, right? The traits that your parents have are most likely going to get passed on to you. And then there's this idea of survival of the fittest. So like we learned, species are constantly competing not only with each other, but also with other species. So the species that does the best and has the best traits are going to win. The classic example of the giraffe. Giraffes have evolved long necks to eat the leaves off the treetops, right? If you're a giraffe with a small neck, you won't be able to survive and compete with that food source. Then you'll eventually die off, and giraffes with longer necks will survive into the next population, in the next generation. So again, these three main ideas can be boiled down to one sentence, that is descent with modification. So species have an existing trait. If that trait is beneficial, they'll pass it on. If not, that trait will then be weeded out of the population because the animal will most likely die before they can reproduce. So here in this photo, we see that there's a bunch of different frogs. They're all of the same family, which is frogs. But then they also have all this different color and variation to fit different niches within their environment. So there's three different mechanisms of evolution. Two is an external process and one is an internal process. External processes happen outside of the organism's genes, and that's natural selection, artificial selection, and sexual selection. And then there's internal processes that happen within an organism's genes, which are genetic mutations. So this is an example of natural selection here. Let's just say many, many years ago, there was a lava flow that created really black rock because lava rock is black. A population of mice has then moved into that new area, area of lava rock, and they get hunted on by birds. So mice with tan coats do not blend in as well as mice with black coats. So they're easily spotted by their predators. So therefore, the birds that eat the mice will most likely pick off the tan mice or the white mice because they look a lot different than the surrounding rock, right? So over time, as more white mice get eaten, the black mice survive, and then the population then changes from maybe a 50-50 ratio of black to white mice all the way from only black mice because all the other white mice have been eaten by the birds. And it's most likely that the black mice then reproduce and pass on their black coat or their black fur to the next generation and so on and so forth. So over time, depending on the environment, they have this pressure of not only their habitat, which is now changed from sand to lava rock, but also the pressure of their predator. And that influences the color of the coat of which the mice have and the fur that the mice have. So those external forces, the habitat and the predation pressure, leads to genetic mutations within the mice themselves. It's very important to note that animals do not choose to evolve. Natural selection is driven by mutations, and mutations are random. So an individual is born with a mutation. If that mutation benefits the individual chances of survival and reproduction, that individual will create offspring and pass on that trait. So it was beneficial to have a black coat compared to a white coat or a white fur because you blend in better with your environment. So nature has selected the fur color, not the organism themselves. And then if your mutation is hazardous, the individual will most likely die before it is able to reproduce, not passing on the trait, right? It's hazardous to have the white coat because you don't blend in as well with your environment, so you're most likely get picked off by the predator. Then there's this thing called artificial selection, which is what we do to dogs when we want to make them look a certain way. We just breed them for their traits over and over and over many generations to get the pugs that look like they can barely breathe and have this really scrunched in nose, or to get the dogs that have the really big floppy ears, right? These are desirable traits that we like to select for and breed for because it makes the most money for the breeders. There's also this idea of sexual selection, which can be seen through male-male competition or female choice. In this case, you have elephant seals. Males have a harem of females that they breed with. They most likely pass on their traits. It's oftentimes that the biggest elephant seal is the one with the best traits. It's the biggest, it's the strongest, and it has to defend its territory from other males who are rivals. And so in this idea that the females want to mate with the biggest and best competitor, the male then guards that harem, and then he passes on his genetic mutation to the generation after. Other words, there's female choice. This is often seen in birds where males will put on a pretty elaborate display of their feathers or dances to entertain a female. In this case, in the female's eyes, the bird with the best dance or the best colors has the best advantage in that environment, so they want to mate with that best male there. So sexual selection can be defined by male competition or female choice. That's all I have for you guys today. Is there any questions on evolution? Anything at all? We got one question here. the first slide. This one? Is this the one you want? Or this one? This one? Cool. Yeah. Like take notes on that. Remember, again, these are going to be posted. So I will post this YouTube lecture as well. Okay, so next week, you've got, while Bianca looks at this, I'll talk to you about your assignments. Next week, you've got 3 assignments that are due. You've got the algae beads, you've got an iNaturalist, and the last one is your 6-week, your 6-week ecosphere project. So the algae beads selfie is just a setup with your algae beads packet. By now you should have all gotten the algae research supply packet in the mail. So hopefully you can start with that. I'll demonstrate on what that actually shows today and how to set it up. You want to send me 4 more iNaturalist observations, and then your 6-week ecosphere report. Last week we talked about how to set up your hypothesis. This week I want to see your hypothesis, your independent and dependent variables, as well as any confounding variables, a summary of how you've been taking the data, and then a data sheet that you have thus far. Just go ahead and throw that on a Word document, and that'll constitute your 6-week ecosphere report. So these 3 things are due next week. And I put those assignments on Canvas as well. So if there's no other questions about any assignments that are due next week or today's lecture, I'm going to go ahead and give a little demonstration of how to set up your algae beads. And I'm going to post a PowerPoint that shows the step-by-step instructions, but I thought I'd just show you guys today just to demonstrate and make it easy for you guys. So everyone should have gotten a beaker bag, right? This little thing, this pouch, they're actual little algae in the tube here. And then a few other things, some salts, the algae culture nutrients in this little vial, and then a Secchi disc. So for next week, what you want to do, and from here on out for this experiment, you're going to get your beaker bag, and you want to fill it to 300 milliliters. There's a little thing here on the side that shows exactly how many milliliters there are in this bag. What you want to do is you want to fill it to 300 milliliters. Get your bag. It's best to use filtered or distilled water, but I just use tap water because I didn't have any at the moment. You can use water from a Brita, that's fine too. Go ahead, fill it up to the 300 milliliter mark. Once you've got that, add in your salts and your culture, algae culture. So go ahead and add that in. And then once these are added, try not to spill. Close the bag. Give it a good little shaking. You can even mix it up if you have a little, I think there's a little like stir kit in the bag as well. Stir it around, make sure everything gets dissolved for the most part. And then fill up the rest to the 500 milliliter to the top of the bag. Not quite there, but that's okay for this demonstration. So you've got your bag full of the salts and the algae culture. Now, before you put your actual tube of algae into the bag, you need to pour out 100 milliliters and save it for the next few days. So go ahead and pour out, 100 milliliters or so, and save this. Seal it with a cap. Save it for 3 days because you're going to be using this. This is where all the algae nutrients are in. You're going to pour it in 3 days later. So now that you've got your bag full of algae nutrients and salts, go ahead, take this tube, shake it up a little bit, make sure it looks nice and green. Open up your algae and just pour it into the bag. And this is where the algae will live for the rest of their life. So go ahead and seal it and put it in a dimly lit spot. You don't want to put it in direct sunlight. It's definitely going to be out of direct sunlight. Even just regular light bulbs is fine for it. They'll live. But this is where they'll live for the rest of their lives. So what you want to do is, and to collect data on this, as the algae grow, it's going to get more and more cloudy. So you won't be able to see through as much because they're growing and reproducing and multiplying over time. So how to measure that is you take this thing called, well, first, any questions on the setup of this? The instructions are also in your lab manual, and I'll post a PowerPoint detailing the step-by-step instructions. But any questions right now? Cool. So like I said, as the algae grow and multiply, the water should get darker and darker and darker. We're going to be taking data on exactly how dark the water gets with this thing called a Secchi disc. So in your algae packet, you should have this little measuring stick here. At the end, you have this little disc-like thing. Bend it 90 degrees so that it looks like an L, like this. Can you guys see that? And what you want to do is you want to stick this into your packet all the way down until you can't see the disc anymore, right? Because the algae is super cloudy. By the time you reach in, maybe it's like 3 centimeters down or whatever, you want to look at it. You're going to stick this actually into the bag. I can't do it right now while I'm showing you this, but stick this all the way into the bag until you can't see this little disc anymore. All right. And then whatever number that corresponds to on the Secchi disc, you can see that it corresponds to, 110, 100 centimeters, millimeters, whatever that is. That's the number you record. All right. Does that make sense? So there's also a video on YouTube that I've posted, or that I'll post in the PowerPoint as well. So whatever number your disk disappears, that's what you're going to be recording as your data. The very first day you make your beaker packet, you're going to record data 3 hours after, 6 hours after, and 12 hours after. And then after those 3 initial ones, every day you'll record it for 12 days. And then you will input the data on page 49 of your lab manual. So you'll have this little data sheet here that you'll record the data. All right. So it'll be the Secchi disc measurement and then the day you record. All right. Any questions on that? So next week, the whole data is not due. It's just a selfie with your actual setup in your packet. All right. Any questions?
For the PowerPoint slide, could you make the YouTube video like a link? So
yeah,
because like last time I couldn't copy paste it.
Just
like the link to YouTube, like YouTube and then slash.
Whatever. Okay, I'll throw it into the PowerPoint before I post it.
Okay, thank you.
Yeah. Cool. Any other questions? Then you guys are good to go.
For the data on the ecosphere, do we